Today we're going to talk about muscle contraction. I thought we'd first review our myofilaments. We'll start with our thick filament or our myosin. If you recall, our myosin is our thicker filament or our thicker protein. On that protein, we have some cross bridge heads, which eventually are going to come up, make contact with our actin filament. Our actin filament is the thinner of the two proteins, if you remember from earlier. And actin has a couple parts to it. First, the actin molecule is kind of like a, uh, excuse me, string of pearls. And if you take a look at all the kind of beads, they kind of corkscrew around each other. And this is going to be a pretty important part of the whole muscle contraction part. If you take a look real close at this picture, you can see on each pearl there's a little purple active site that's being covered up right now and blocking the cross bridge head from coming in contact with that active site. The crossbridge head from the myosin actually needs to come in contact with this active site on the actin. A couple other parts of the actin molecule, if you remember, we have the troponin complex. Troponin complex, you kind of think of like maybe the head of a spoon. Um, on here, that troponin complex is going to come into play a little bit later as we have calcium that's going to come attach to that troponin complex and actually change its shape. Next, we have the tropomyosin complex, and this is kind of another protein that kind of corkscrews around with the actin. And the tropomyosin complex is actually what covers up the active site on the actin. So we have our neuromuscular junction. The neuromuscular junction is a motor neuron which comes in contact with a muscle fiber. And so we can have multiple junction sites with this motor neuron coming in contact with the muscle fiber, but it's called the NMJ or neuromuscular junction, and that's the site where these two come in contact. And if you look closely, specifically right here, they don't even actually touch each other, physically touch each other. There's actually a space that is in between the neuron itself and the sarcolemma of the muscle, and that's called the synaptic cleft. Another name for the neuromuscular junction is the synapse. So if you read or hear about that, it's still talking about the same thing. So we have our motor neuron. We got our motor neuron end plate. That's the sarcolemma here that has all kinds of surface area. And on the sarcolemma, we have all kinds of sodium channels that will be open or closed. In case of a relaxed state, they're going to be closed. And we're going to need to get those open to generate a new electrical signal to actually have a muscle contraction. Inside the neuron, we have synaptic vesicles. You can see those made by the Golgi apparatus. Those synaptic vesicles, you can see all kinds of them in this particular neuron, house a neurotransmitter. A neurotransmitter is just a chemical in a neuron that is going to be released to cause some particular action to take place. So specifically, the neurotransmitter we're going to be focusing on is acetylcholine. Now, acetylcholine can do a number of things. We're going to obviously focus on the muscle contraction part of acetylcholine. Next, you have to define a motor unit. A single motor unit is when a neuron interacts with muscle fibers. And so we can have all kinds of muscle fibers controlled by a neuron or just a few muscle fibers. So as few as 4 muscle fibers, single muscle fibers, can be controlled by one neuron, or thousands of muscle fibers can be controlled by a single motor neuron. And so the difference is how much power or how much control do you need. So for example, maybe we want to fire one neuron and that fires our gluteus maximus or our pectoralis major muscles, and we generate a lot of power. one neuron sending a signal to lots of muscle fibers to have a muscle contraction, or maybe it's a situation like in our fingers where we have lots of neurons firing at different times to control movements of our fingers so we can grab something or move very precisely. And we have maybe closer to a 1-to-1 ratio there. So here we have an animation just to kind of lay out where we're headed, and I'll explain a little more into detail.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholine esterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
So let's see if we can't break a little bit of this down. We had our acetylcholine in our vesicles, in our motor neuron. We get an electrical signal. You decide you want to move some part of your body. Maybe you want to walk. Maybe you want to pick up your sandwich to take a bite of it, whatever the case may be. And so you send a signal from your brain, an electrical signal, down our motor neuron. And you can see that traveling down this way. And inside, we have our vesicles that are filled with the neurotransmitter acetylcholine. Keep in mind, as that electrical signal comes down, that calcium now comes rushing in again, causing the synaptic vesicles to go through exocytosis and fuse with the membrane of the neuron. The acetylcholine then is going to get dumped into the synaptic cleft here, and it's going to diffuse from high concentration to low concentration, and it's going to attach to the receptors on their sarcolemma. Remember, our sarcolemma is invaginated like this, and we have a lot of surface area, so we have a ton of receptors that the acetylcholine is going to come in contact with to now open these gates, these sodium channels, so sodium can come rushing in. Once the sodium comes rushing in to the muscle cell, we start generating a new electrical signal, and that electrical signal is going to travel, we'll come down here, that electrical signal is going to travel all throughout the sarcolemma, okay, up, down, including deep into the muscle fiber itself. Remember, we had our transverse tubules that go deep into the muscle. Well, that electrical signal will transmit right down that transverse tubule. And remember, we have our triad next that includes our transverse tubule. Two cisternae, which is part of the sarcoplasmic reticulum. So we have our transverse tubule and two cisternae that were right next to it. And then in those cisternae, we have some more calcium that's going to play a role in our muscle contraction, as we'll see here in a little bit. Now, first, your book talks about myasthenia gravis syndrome, which is a disorder where the receptor sites on the muscle fibers are compromised or broken down or destroyed. And so if we don't have the receptor sites on our sarcolemma, then we can't have a muscle contraction. So the acetylcholine, once it's released, it's got nowhere to go, or it has fewer places to go. And so a person with this particular syndrome experiences fatigue, weakness, a whole bunch of different scenarios. All right, so just to give you the macroscopic idea here. So here's our muscle fiber that comes down. We got electrical signal coming down. We got a vesicle inside us. That electrical signal comes down. Calcium comes rushing into the terminal end. The vesicle fuses with the cell membrane of the neuron, releasing its neurotransmitter acetylcholine. Acetylcholine now is going to diffuse across and attach to the receptor, and we're going to generate a new electrical signal. With the sodium channels open up. We'll get to that a little bit later. That electrical signal is going to travel down the sarcolemma, including down the transverse tubule, deep inside the muscle. And the electrical signal is going to travel that way and around the backside and down each one of these transverse tubules, causing the transverse tubules that come in contact with the cisternae, causing calcium to be released. Now, where'd the calcium come from here in the muscle cell, do you ask? Well, it's stored inside the sarcoplasmic reticulum and in these cisternae, these big holding tanks. And so the calcium gets released. Well, the million-dollar question is, what does the calcium do? Glad you asked. Recall, we have our actin filament, our little strands of pearls that are corkscrewed around each other, our troponin and our tropomyosin complex. Here's our troponin, that's the spoon, and then the tropomyosin, the spoon kind of attaches to that other protein that continues corkscrewing around the actin, covering up that active site. Now, we can't have a muscle contraction if the cross-bridge head from the myosin can't come in contact with that active site. We can't have a contraction. So calcium gets released from the sarcoplasmic reticulum. So here's our calcium that's being released, and the calcium actually attaches to our troponin complex. It comes and changes the shape of that troponin. Remember, troponin is attached to our tropomyosin, so now it's going to cause all that troponin tropomyosin complex to kind of rotate a little bit, and you can actually see the active sites are now exposed. Now we can have a muscle contraction. These cross-bridge heads on the myosin can come up and make contact. And so you start to see that in a progression down here, down below. Here you can actually see the tropomyosin or the myosin cross-bridge head has made contact with that active site. And then we can use ATP to ratchet in the actin. And so we got two sides of this, if you remember the slide earlier. We got cross-edge heads pulling from the right side in, and cross-edge heads pulling from the left side in. And that's how we get a muscle contraction. So here you can see the actual Z lines are going to shorten. And so this is what we call the sliding filament theory. Here you can see myosin, and the darker purple here are the cross-bridge heads. And when those cross-bridge heads are allowed to go up, here you can see we're at a relaxed state, so they're not in contact. Here we got partial contraction, here we got full contraction, and so those cross-bridge heads are allowed to go up and make contact with the active sites on the actin, pulling the actin filaments in. So what happens to our Z line from Z line? Our sarcomere actually gets shorter. So our H zones and our I bands actually shrink or get narrower, and those Z lines move closer together. Remember, a muscle contraction is when the muscle fibers shorten. And so now we have our muscle contraction.